Hello, everybody, and welcome to Scripps Research, fully interactive resources for students and teachers or FIRST. My name is Rosie Alvaran-Seckler, and I will be your moderator this afternoon. For those who are joining us for the first time, Scripps Research is one of the largest nonprofit private biomedical research institutes in the nation, and our mission is to develop the next generation of medicines and diagnostics to address the most pressing health problems in the world, such as COVID-19. For those who are joining us again, welcome back, and welcome to the new summer series, Careers in Biomedical Research. As I mentioned in previous webinars, our goal is to expose some of the different career paths available to students who are passionate about biology and chemistry and curious about research, especially in an academic setting. We hope you can get some insights from the daily work lives of our scientists and their journeys to where they are now. Before we move into our presentation today, I'd like to remind everybody of a few housekeeping items. All participants have been muted and your videos have been disabled. The format today will consist of a 15-minute presentation by our speaker, followed by a 10 to 15-minute Q&A session live, where we're gonna be taking your questions. And with that, I would like to introduce our speaker. Today's speaker is Pierre Bayerjon. He's a senior robotics engineer from the lead identification group at Scripps Research on the Florida campus. Prior to joining Scripps, Pierre earned a Master of Science degree in computer engineer from Florida Atlantic University. And over the past 15 years at Scripps, Pierre has supported academic drug discovery efforts by developing, assembling, and integrating novel automated software and hardware platforms. And today he will be talking about the application of engineering and robotics to drug discovery. Welcome, Pierre, and thank you.
Hello, thank you very much for the introduction. Switch over to my slides here. All right, we're able to see the slides that I've got up now?
Yes.
Okay, perfect. Thank you very much for the introduction. I'm happy to have an opportunity to talk to everyone today how we're using robotics and automation in the drug discovery effort here at Scripps Research. So I'd like to start off with a little bit of an abstract view of drug discovery as a general concept. So if we were to look at what it takes to go from a idea or an experiment, a project in a lab, all the way through to getting one FDA-approved medicine that a doctor can actually write a prescription for and prescribe to you, that process takes somewhere in the order of 10 to 15 years. That includes all of the laboratory research that occurs in a facility like we have here at Scripps Research. That includes the FDA clinical trials. You may have heard before of phase 1 clinical trials 2 and 3. Ultimately, once you pass through that battery of clinical trials, you can get onto an approved medicine. So it takes quite a long time to get a new medicine actually approved once you've started a project. So not only does it take quite a bit of time to get a new medicine to market, it's actually quite expensive as well. So you can see here over the course of many decades, the cost of a new drug has increased. So as of the early 2000s, it cost about $1.2 billion to bring a new drug to market, from the early lab stages all the way through actually getting it to where a doctor could prescribe that medicine to you. So the question is, how can we reduce the timeline so that it doesn't take quite so long to get that new medicine out? And how can we also reduce the cost? So some of you may have seen a show like this before, like CSI, or maybe you've worked in a lab before and you've seen someone with a pipette at a benchtop and they have a test tube. And inside of that test tube, they're essentially performing one single experiment. And with this experiment, they can determine some results about some biological or chemical process. Now, what we're doing in our lab is we kind of take that same idea of performing an experiment, except instead of in a test tube, we use specialized pieces of plastic labware called microtiter plates. So you can see in the top right corner of this slide, there are an image that contains 3 plates. So there's different densities. You see here a 96 well microplate, a 3D4 well microplate, and a 1536 well microplate. Now, 1536 well microplate means there are 1,536 experiments that can be performed simultaneously in that microplate. And what you'll also notice if you look at the microplates, you see what we call wells. They're these little round circles or little square outlines, and that's actually essentially each one of those wells is an individual test tube. And so if we were to take a 1536 well plate and put it under a microscope, you would see what you see in the bottom right-hand corner of this slide here. So you see two wells side by side. These wells happen to have some cells in them. The cells have been dosed with some different drugs that are in our compound library, and you can see two different responses. In one of them, the cells have a blue color, and in the adjacent well, a green color. So we're able to perform these experiments in these very small wells, and by reducing the amount of reagent that's needed, we're reducing the cost. And then because these plates are designed in such a way that they can run on a robot, we're able to run these microplates very quickly through our system. So not only is the density of these plates such that you can have many experiments per plate, but our robotic system can actually run hundreds of these plates per day. So in the course of a day, we can run hundreds of thousands of these small experiments, upwards of 1 million as well. And these robots, they don't get tired, right? So they can run 24/7, they can run through the night. And all they need is a robotic operator that can basically explain to them in the robot's language what the experiment is that's being run and how that experiment should be performed. So what does our lab actually look like here at Scripps? So we're doing what we call high throughput screening. And high throughput screening is really where you have a couple of different specialties coming together. So you have chemistry and biology meeting with robotics. And the idea here is that by combining these things, we can take all of the advantages of robotics in terms of the speed and the reduction in cost, and we can reduce a lot of the manual task that's required to perform some of these experiments. So in the top image here, you see a part of our lab that we call our compound management or sample management area. And that's where we are preparing large compound libraries to be tested on these robots. Now, these compound libraries consist of hundreds of thousands of compounds in most cases. And what will happen is we have an experiment, and each one of those compounds in that compound library will be tested in that experiment. And it's essentially, you can think of this as a needle in a haystack approach. So we're attempting to find out which items or which compounds in that library give us some result in an experiment that we can then follow up with as well. In that top image on the right side, you can see what we call our assay development area. So that's where our biologists work, and they're working to get these experiments in a format that can actually be run on the robotic platform in these microtiter plates that I showed you on the previous slide. On the bottom image here, you can actually see our robotic laboratory. So there's two main robotic work cells in our lab. The one on the bottom left, we call our high throughput screening system. And on our bottom right, we call it the hit picking system. So the high throughput screening system actually runs or executes those experiments. And the hit picking system, once we get the results from the experiments, can actually go back and do follow-up work to rearray or pick out samples we want to perform additional testing on. So to give you a little bit more of a concrete view of what this robotic platform looks like, I've got a video here that I'll narrate. So this is one of the robots we've got. This is the high throughput screening system. And all this robot does, its only purpose in life is to shuttle these microplates around to different devices that are installed around the robot. That's all it does. You can kind of think of it as a conductor, and there's an orchestra, and the robot is the conductor that's telling these devices what to do, how to do them, and it's going around and giving these plates to the devices to actually perform the work. So here you can see the robot picking up a plate from one of these devices. We call this our pin tool. It'll put lids on them. We can control the temperature and humidity. So we've got quite a few sophisticated components on this robot that allow us to complete the experiments any way they're needed. And one benefit of the robot is you can really have many things going on. So while one device is running on one side of the robot can bring a different microplate to a different device and run things in parallel. So that's one of the ways we're able to do things very quickly. And in this case here, what you're seeing is a device called the flying reagent dispenser or bottle valve dispenser. So you can actually see a line with some red liquid in it. Those are actually cells that are suspended in media. And what it's doing is it's using a valve similar to what you might find in an inkjet printer to actually dispense very small volumes of those cells into each well of the plate to perform the experiment. You can see here another example of while that dispense is occurring, the robot has gone off and retrieved another plate at a different device, and it's working through this kind of sequence of steps that it needs to run every plate through in order to complete that experiment fully. And at the end of the day, once the plates have completed on the robot, all that information gets put into a large database. That way our biologists and our chemists that work at Scripps can query that data and do follow-up work on it as well. So that's kind of an abstract view of the drug discovery process, and more specifically what we're doing here in our lab. But what does it actually mean to be a robotics engineer in our lab, and what do we do? What's the job function? So certainly we work with robots and laboratory automation. So you see here an image of one of our large robots on our hit picking system. Now, not everything for laboratory automation involves a large robot arm. There could be plenty of small benchtop devices, anything that basically lets a user step away from some work and it will perform the work on their behalf, is something that we use and we like to leverage in order to make sure that we're reducing any kind of opportunity for error or any kind of manual process that's occurring in our lab. So our robotics engineers are configuring, repairing, and running laboratory automation, both the large systems, our benchtop systems. Spend a lot of time as well developing software and custom electronics and administering databases. So you can imagine that as we're getting all this data out of these systems, we have to curate that data, perform quality control checks on that data, and make sure that everything is appropriate before we report it out to anybody. So all of these kind of job functions you can think of as being related to certain academic areas of interest. So maybe you say, this sounds interesting. I'd be interested in doing some kind of work like this at some point. What should I be studying when I'm going to school? So the areas that are related to this that I would recommend investigating are chemical engineering. So we've had a number of our robotics engineers that have worked in our lab have backgrounds in chemical engineering. Another one is computer engineering. So that's what my background is in. Certainly computer science as well handles that aspect of the software development. And then you've got things like electrical engineering and information science for dealing with a lot of the electronics and just the large volume of data that we're dealing with in our databases. Okay, so I mentioned I've got my background in computer engineering. So how did I actually get here? How did I land as a robotics engineer working at Scripps Research? So I graduated from Florida Atlantic University in Boca Raton in 2004 with a bachelor's degree in computer engineering. A year later, I received a master's of science in computer engineering. And the common theme with my university coursework was an interest in working with robotics. So anytime I had an opportunity to select a project that I wanted to work on with a class at FAU, I always wound up doing something with robotics or automation. So that includes, if we go from the bottom left to the bottom right, starting out with a small robotic arm that was able to do image processing to automatically identify and select objects based on their shape and color. After that, I had worked on, for my undergraduate senior project, an automatic harmonica player. So we took an old printer and we mounted a harmonica on it. And with some custom electronics and custom software, we were able to actually get the harmonica to play in an automated fashion, any kind of music that you're interested in. And then at a graduate level, I managed to reuse that robot arm from the image processing class, mounted onto an air hockey table with a little bit of MATLAB. So many of these different projects use different technologies. One that happened to be prevalent was MATLAB quite a bit. And that was able to actually play air hockey in an automated fashion based on the image that you see there. And one system that kind of just coincidentally lined up with the work that I would end up doing ultimately here at Scripps was a graduate course that I took on robotics. So Motorola used to manufacture cell phones down here in South Florida. At some point that they ceased operations with the manufacturing of the cell phone lines, and they actually donated some of their systems to FAU in the robotics lab. And one of those systems happened to be a print and apply system. So these cell phones would come in on a pallet, and they needed barcodes to be tracked through the manufacturing process. And this robot would do that. So cell phones would come in and it would go pick up a label and put it on the cell phone, and then the cell phone would continue down the manufacturing line. We actually reverse engineered this robot in our robotics class. Instead of taking labels and putting them on the cell phones, we put 96 well plates on a pallet, and those pallets with the plates on them would come in, and we could actually pipette 4 wells at a time with the system. So it just happened to be coincidence that we had chosen this as our project. And when I happened to interview Scripps after I was done at FAU, I showed them this, and they said, "Oh, well, let us show you the pipetting robots that we use. They're a little bit more sophisticated than this here." But this is kind of give you an idea of what the framework was and what kind of projects I was working on before I wound up working at Scripps Research. So that's how I got here. Obviously, you've seen the robots in the video in the lab. And that's a big part of our daily job, is working with those systems. But there's also a question of what else can we do in addition to using the existing technology in our lab to kind of continue improving the processes. And a big part of that recently for us has been leveraging open source technology. So I'm sure many of you have heard of the Raspberry Pi. It's a small computer. Also the microcontroller called the Arduino. There's other technologies that have become much more accessible recently, such as 3D printing. And so I've got some examples on the bottom of this slide here. So we've come up with custom incubator shelving that we've 3D printed and manufactured, and we put these custom shelves onto our robotic platform to allow us to do some novel biology around 3D spheroids. So that's something that would have been very difficult to do 10 or 15 years ago without going to a custom machine shop and having something very expensive fabricated for us. Now we can do all these things in-house. It's really empowered our staff to continue to learn new tools and learn new technologies, and then come up with things like this. I mentioned the Arduino and the Raspberry Pi. So you see on the bottom in the center there, a system for automatic quality control of liquid dispensing on our robot. And then we've created a wide variety of electronics projects and 3D printed fixturing around the lab that's been quite helpful as well. One other kind of concrete example of how we're using open source technology to have an impact in our lab is this Orbital Shaker example here. So sometime last year, one of the biologists had a project come up where they needed to take an orbital shaker and have a flask that could be put on there, and that flask and orbital shaker needed to operate in a humidified incubator. Now you can go out and you can buy commercial orbital shakers. They're fairly expensive, starting generally at about $1,000 a unit and going up from there. There's a couple of issues though. They're generally fairly large and bulky. And if you're putting something like this in a humidified environment, there's a good chance that there's going to be some kind of corrosion or failure due to the amount of humidity if that device hasn't been specifically designed to operate in an environment like that. So we looked online and we found what's called the DIY Bio Orbital Shaker. It's completely open source. You can kind of see the components laid out there in the top center image on this slide. It costs about $100 in components to put one of these together. And the great part here is we saved a bunch of money doing this, but now if there's any issue due to humidity or due to anything else, or if we wanted to modify the function of these, because we put them together and build them ourselves, we had complete control over how they functioned and able to service them very easily as well. If something broke and we needed a new part, we didn't have to call up a vendor and have a part shipped. We could just 3D print a new component as needed, or order a new motor. Another benefit here of having an open source system like this, we have maybe a need for putting other things on the orbital shaker other than flasks with cells in them. Maybe we want to put some conical vials or some other shaped labware on there. And because we have the designs for this, we can very easily create custom mounting brackets that can shake a wide variety of objects as well. So we've been using these for a little bit of time now, since October of 2019. They've been working quite well. So that's kind of another example of something that we're applying, our knowledge to and continuing to learn in the process as we go. So one of the questions that was posed to me as I was getting this presentation together was what keeps me motivated and coming to work every day and being excited about what I'm doing. And our lab, the high throughput screening lab here at Scripps, has been in operation for almost 15 years now. And one of the first projects that we ever worked on in our lab was a project called S1P1. This project ran on our robotic platform in August of 2006. And in March of this year, one of the compounds that was discovered when it ran on our robot actually got through the process and became an FDA approved medicine. So if you were to go on Google and you search for a drug called ozanimod, you'll see that Bristol Myers Squibb has actually licensed this compound, and it's now available for a prescription for multiple sclerosis. So that's actually quite been a neat experience to see what that's been. It's certainly taken 14 years, so you have to be quite patient in seeing the kind of results and impact of your work. And there's plenty of projects that we run that don't get into approved medicine status. And certainly our team was a very small part of the overall process as well, but that's been very motivating to have had an opportunity to be a small part of getting a drug that's been approved for use in man now. And then one of my final slides here I'd like to mention is, we're very fortunate to have an opportunity to work in a lab like we do here at Scripps. And so it's very important to us to give back to the community and engage the community and kind of talk about what kind of research we're doing. That includes doing things like the Palm Beach Mini Maker Fair. We've organized through the Scripps Education Outreach Department a variety of outreach efforts that includes, working with local educators to come up with curriculum in the classroom based on robotics, Lego Mindstorms, and things like that. And we've also hosted a wide variety of science outreach events as well, where we try and interact with the community and really have an opportunity to discuss what kind of research we're doing here and what the impact of that research is outside of Scripps at large. So with that, I'd like to say thank you very much for your attention. I'm happy to take any questions you may have. If you happen to think of a question after the presentation here, I've got my email address up there. You can send me an email. If you'd like to know more about the research that we're doing in our lab, you can go to the link that's shown there as well, and you can find videos and academic papers about some of the other research that we're also doing. Thank you.
Thank you so much, Pierre. That was a great presentation. I see that the students are motivated. We already have 4 questions in, which is early normally for webinars. So I'll go right into the questions. So in the beginning of the presentation, we got the first question, and I think you were discussing cost of drug discovery. And so the question is the cost increasing because of inflation or other factors?
That's a good question. I'm not sure that I can give a real detailed answer on that. I know that one of the large portions of the cost is actually that FDA approved testing. So I'm not sure exactly what percentages of the cost is, the equipment becomes more expensive due to inflation or something else. I do know that from what I've heard, the large component of that is actually that FDA approved testing. Once you start trying to put some new compound into any kind of individuals, there's a lot of safety that goes around that and making sure that these drugs are not only doing what they're supposed to be doing, but that they're not having any adverse impacts as well. So my understanding is that's where actually a lot of that cost occurs. But anywhere we can save in the process is certainly still a benefit. So that's where we try to leverage that with the robotic platforms.
Okay, great, thank you. Then the next question is, do you use a proprietary software? What are the primary coding languages used?
Sure, so that's a great question. So I would say that almost all of the systems that we have in our lab that were commercially available are kind of closed proprietary systems that we don't necessarily do any programming work on. A lot of the software development that we do in-house, we spend a lot of time developing web-based tools to interact with the data that we're generating. So we use technologies like PHP for developing interactive web pages that can query Oracle databases or MySQL databases. In terms of what programming languages are best to learn or are most interesting, I would say it's really project dependent. So there's quite a few programming languages out there, and depending really on, so you saw on one of my slides there about the air hockey table and the robotic vision system. I would say that the technologies or languages to learn are those that will help you accomplish some specific task. So instead of randomly choosing a language to learn, first choose a project to work on, which is, I find that in my experience, project-based learning is very impactful. So generally, if you choose something that you want to accomplish, and then you determine what tools will help you accomplish that task, and you can find online, I want to find a programming language that's good for web development, or I want to find a programming language that's good for embedded system development. So as I mentioned, the first thing I would do is choose a project or something you want to work on, some problem you want to solve, and then find the tools for that problem.
Great, thank you. I think this is kind of like a related question, and a question that I've been getting a lot from students. So what coding languages and software do you use when making these college projects? I know that you just touched on some of those, but there's R, there's Python. So so if they learn one of them doing what you just suggested, with the project, is it easy to learn a new one or to translate it into another language?
I think so. Certainly there's differences between programming languages, but many of the core concepts are the same. And one of the great things that students have access to now that I didn't necessarily have access to when I was going to school is there's a ton of great resources online that will help you learn different languages or tools. So for example, you can go on YouTube and you can find programming tutorials. You can find examples of specific projects that people have worked on. And so those things will help you transition. If you learn one thing, let's say you learn C or Java, and you now want to go to Python or something else, you can generally find websites that will even help you transition from one to the other. They'll show you what the differences are between those languages and what things you might need to focus on as you're trying to learn something else.
Okay, great, thank you. Next question, how long does it take to code and construct a robot used in a laboratory setting?
So as I mentioned, most of the robots we've got are commercially available, but we have actually implemented some custom robotics, mostly for our education outreach efforts. And it's really dependent on what functions you are looking to have those robots do and what software libraries or software programming interfaces are already available. To give you an example, we have a robot here that we use that can essentially transport microplates from one location to another. It's a fairly small robot, and it came with an application interface that basically lets you say, take a plate from one location to another. So programming that's fairly easy because you don't have to worry about how do I program the motor in the robot? How do I program some other electronics interface? So it's kind of dependent on what you're programming. I would say though, depending on that, like let's say for example, some of the projects I worked on at school that were MATLAB based, those were all done over the course of one semester. So generally those were on the order of a couple weeks. Again though, those were systems that came with a platform where you didn't have to start from scratch every time. There was already some underlying technology framework there that you could build on top of to build the thing that you're interested in doing. So it's kind of complexity dependent. Could be a couple of weeks, could be a couple of months, could be much longer than that as well.
I guess as a segue on that question, how is it possible to use something as bulky as a robot to perform such precise tasks like in the lab tests?
So that's a great question. So these robots that we use, you actually see in this picture here, these are Stobly robots, and they are, so the group that designed this robotic system that we got actually came out of the automotive manufacturing world. And these robots are very accurate and precise in placing these microplates. And so the robot itself only has to worry about where it's putting the plate down on one of the devices that surround the robot. After that, it's the job of the device to worry about the accuracy and precision of making sure that it's working in the correct place on that microplate. That said, there are kind of tricks around that. So for example, when this robot brings one of these plates to a device to have some work done on it, typically what will happen is it'll put the microplate down, and then there's some locking mechanism that will engage and actually put that plate in a very specific location that's been pre-aligned and predetermined. So there's enough slop there for the robot to get in and handle the plate, and then afterwards it can be locked into place with some active positioning units.
Great. So the next question is, what sort of resources do you use to find open source lab automation, and what forums do you post your inventions on?
It's a great question. So I would say a great place to start is somewhere that has a code repository like GitHub. You can certainly search there for terms of different things you're looking for. Some of the online repositories for 3D printing are great for this as well. So for example, Thingiverse, the NIH actually runs their own 3D print exchange where you can find some of these devices. In terms of professional publications, there's the Journal of Laboratory Automation that we've published some of our resources in. There are also video-based journals that you can find online that could be helpful as well for actually seeing what's happening, as opposed to just if you don't want to just read a journal article. And then generally just searching on social media. So you can find people posting their projects on Twitter, on Reddit. There's certainly dedicated subreddits to electronics and programming and certain things like that. So really there's a wide variety of places that you can find this information, but we publish most of the time in some of the technical journals that are associated with automation in drug discovery.
Great. We have a question here about internships, and I'll post my email address on the chat so everybody who has a question about internships can email me. But I know that UPR specifically, every year you host many of our interns. So can you talk a little bit about what will be the experience of an intern in your laboratory and what kind of projects they have?
Sure. So typically, when folks come through our lab for an internship, we do a boot camp first. So a lot of the interns that work in our lab have never worked in a science environment like this before. So typically, the the process is we teach some of the basics of handling microplates in the lab, how to pipette in microplates, how to use those microplates to perform experiments, what type of experiments you might be able to run on some of the plate readers that we have in the lab, different chemistries that can occur in there. Then we also do basics, so learning things like Microsoft Excel is very important because once, as I mentioned, with these robots, they can generate a lot of data. But even if you're not using one of these large robotic platforms, something like Microsoft Excel can still be very powerful to learn, even for handling small volumes of data. So we'll go through not only the wet chemistry lab training, Excel training, and then once we're done with the initial training with those kind of tools, we'll typically have some kind of project set up for helping to improve a process that we have in the lab or help us better understand some technique or something like that. In most recent year here, we had someone come through our lab who was helping us perform quality control on small dispense valves that we use in our lab. These valves are fairly expensive. And they could fail. And so they were helping us come up with a way to help us clean these valves so that we could reuse them on the robotic platform. And then, things like that. So.
Great. Another question just came in. How do you stay up to date with the trends and the developments in the biomedical engineering industry?
Okay, that's a great question. From a professional perspective, that involves going to conferences, going to some of the technical presentations there, following along with industry journals, and then also any kind of online publications as well. Social networks can be used for that as well. So I mentioned, Twitter and Reddit, LinkedIn can be good for that as folks that you meet over time can post some of their professional accomplishments and what their discoveries are there. So I wouldn't say that there's one particular place, but generally finding the professional conferences or the professional publications in the area of interest that you have will be a good place to kind of lead you into what the latest things are in that area.
Right. Well, I give a chance for people to add any last minute questions. I guess I'll go ahead and ask. So as you know, I go around colleges doing recruiting, and some of the questions sometimes I get from biomedical engineer major students or engineering students is how they can translate their majors into maybe not only a lab like yours, that is the niche is very specific, but into any drug discovery laboratory. So what advice do you have for those students?
So my advice is if you have an opportunity, uh, find some coursework that you can do, or it doesn't have to be coursework, but find something that has a project-based component that you can use as part of the kind of story you tell for your educational experience. So for example, with the projects I worked on, um, when I was doing my undergraduate and graduate work, I was able to document those. And then when I interviewed here at Scripps, I basically had a portfolio where I could say, these are the things I learned, and this is how I applied the things I learned. So that's kind of the value for me in showing not only did you take a course and get a certain grade in a course, but you're able to actually show how you were able to utilize that knowledge for something concrete and show what you did with it. So that's why I'm a big proponent of project-based learning. So if you have an opportunity, that could be lab-based too, right? Maybe the course you're taking has a lab component and you can show or document what are the things you actually did in the lab. Um, that would be my advice, is find some way to do a concrete hands-on thing that you can document and then show as well.
That's a great advice. And before closing, I guess like for my own benefit, I was listening to the news the other day and they were saying that robotics is the career of the future. And I have young kids. So how young can you start kids into this arena, and what is available out there to get them started?
Sure, so that's a great question. How young you can get them started, I don't know. I've got two daughters of my own, and they haven't hit 2 years old yet, but I think I'm already ready to kick that off. Maybe that's a little too early. I would say certainly things you can engage younger children with, things like Lego puts out what's called the Lego Mindstorms kit, and that's a very easy to use system because you don't have to worry about mechanically fabricating things to hold motors or sensors. They can just be clicked together. Systems like that Lego Mindstorms kit also have a visual programming elements. You don't have to worry about learning lines of code and worrying about what is the command or the function that I need to do to turn a motor on or off. They have like little icons, and you can just drag the icon will say turn the motor on, and you drag that down, and then you connect the motor, and you're off to the races. There are certainly other systems other than the Lego Mindstorms, but I find that the kind of barrier to access for that in terms of the complexity of the system is pretty low. There are other, you can almost go on Amazon and find a wide variety of robotics and programming kits at different levels of complexities. But that would probably be one of the start points, would be something like a Lego Mindstorms kit. Also, there are competitions or groups around the United States, even around the world, called First Robotics. So if anyone hasn't heard of First Robotics, I highly recommend checking this out. It's essentially a competition that kicks off anywhere from grade school up until high school, and they have different levels of competition. So the high schoolers have their own events, the grade school kids have their own events, and essentially what happens is they're given some task, like you need to build a robot that puts a ball in a little basket or something like that, and they have a fixed amount of time, like say 6 weeks, to build a system like this, and then they actually have to go to a competition and demonstrate it. And the great thing here is not only did they build the robot and does the robot do the thing, but they are graded on, how did they document their design process, right? How did they document how their team worked together? How did they do fundraising to purchase the components for the robot? So those things are helpful as well, because maybe not everyone is interested in programming the robot or building the robot. You certainly need people who help come up with a business plan for how is that robot going to be funded, and how are you going to market the robot you're doing, and how are you going to interact with the community to get community support. We've got local First Robotics teams here that operate. I know there's some in Delray Beach and Boca. I'm sure there's some in Jupiter area too. And that would be one of the big recommendations I would have, is if you're interested in this and you don't know where to start, reach out to a local First Robotics team, because they actually, part of their mission is to reach out to local communities and engage the local communities in education outreach and show them what resources they have access to get more involved.
Awesome. Thank you for that advice. So I see that there are no more questions. So maybe I'll give you an opportunity to give your final thoughts, and then we'll close the call.
Yeah, sure. So I'm very appreciative of everyone who logged in today for the presentation and for the questions. Thank you for your time. And like I said, if you have any other questions you think of afterwards, feel free to send me an email, and I'm happy to talk to you more about that. So.
Thank you so much, Pierre. It was a great presentation. It was a great conversation. I think that this is a hot topic, a hot arena. Like I said, it's the careers of the future will involve a lot of these, and students certainly have a lot of questions. This presentation is going to be posted on our YouTube channel and also on our Canvas page. So definitely, I'm sure that more students are going to see them. And thank you for sharing your email address, for volunteering your email address so they can email you. I see a note that they're thanking you and thank you for the great presentation. And so with this, I'll close the call. When we close the call, you will be redirected to our webpage where you can find previous webinars and you can register for the webinars that I just showed on the screen. So thanks again, Pierre, and thank you everybody for joining today, and I'll see you next time.
Bye.